Bismillahirrahmanirrahim. Assalamualaikum warahmatullahi wabarakatuh. First of all, let me introduce myself. My name is Muhammad Afiq Iman bin Hajali. I'm from M3802204C. So today, I would like to present about Transgenic rice plant synthetic CRY2AX1 gene exhibit resistant to rice leaf folder. So, let's be started. So, as you can see here, first we will go through the introduction, and then we go to the method, next result and discussion, and lastly conclusion. So we can start now. So first we will go through to the introduction. So transgenic rice plant expressing with the synthetic CRY2AX1 gene have been developed to combat the rice leaf folder, which is an insect pest that can cause significant damage to rice crops. The CRY2AX1 gene derived from Bacillus thuringiensis encodes a protein toxic to the leaf folder. By introducing this gene into rice plant, scientists have successfully made the plants resistant to leaf folder infestation. This genetic modification provides an effective and sustainable solution to protect rice crops from the devastating impact of the rice folder, ensuring better yields and reducing the need for chemical pesticide. So next, we jump into the method. So we have three methods, which is gene isolation, gene cloning, and plant transformation. So first, for the gene isolation, the CRY2AX1 gene is isolated from the bacterium Bacillus thuringiensis, which naturally produces insecticidal proteins. The CRY2AX1 is specifically selected for its toxicity against the rice leaf folder. Next, for the gene cloning, the isolated CRY2AX1 gene is inserted into a cloning vector, which is a small DNA molecule that facilitates gene transfer and replication. This creates a recombinant DNA molecule containing the CRY2AX1 gene. And lastly is the plant transformation, which is the recombinant DNA molecule containing the CRY2AX1 gene is introduced into rice plant cells. This can be achieved using various methods such as agrobacterium-mediated transformation or biolistic, which is gene gun transformation. The transformed cells are then cultured in a laboratory to regenerate whole plants. So next, we jump to the results. So this is the result that we get, which is 20 putative transgenic rice lines were regenerated and putative transgenic plants were transferred to the greenhouse for molecular analysis. Total genomic DNA from the putative transformants was subjected to PCR analysis with CRY2AX1 and HPTLL gene-specific primers. Out of 20 plants regenerated, 16 were found to be positive for the implication of the 800 BP and 630 BP internal sequence of CRY2AX1 and HPTLL gene respectively. And next, for the discussion, transgenic plants with insecticidal crystal protein for Bacillus thuringiensis could drastically reduce the use of broad-spectrum insecticide against insect pests. However, there is a risk that insects could become resistant to the Bt toxin. Therefore, it is investigated that gene pyramiding in transgenic plants could be possible strategy for simultaneous expression of two or more cryogenesis. To improve the insecticidal property of the cryotoxin, a chimeric Bt gene was developed with CRY2AA and CRY2AC sequenced from the indigenous isolates of Bt. The earlier study proved that the chimeric CRY2AX1 protein expressed in the Bt was highly toxic to the larvae of the rice leaf holder. Lastly, for the conclusion, in conclusion, the development of transgenic rice plants expressing the synthetic CRY2X1 gene have proven to be an effective method for conferring resistance to devastating rice leaf folder. By introducing the CRY2AX1 gene, derived from Bacillus thuringiensis into rice plants, scientists have successfully created a defense mechanism against the leaf folder pest. The CRY2AX1 protein, encoded by the CRY2AX1 gene, is toxic to the leaf folder larvae, causing damage to their gut lining and leading to their damage. So, I think that's all from me. Thank you.